and thank you all for being here today. So today I'm going to talk to you about modeling malaria development in mosquitoes and sort of trying to answer this question of how fast can mosquitoes pass on the malaria infection. And so I'll give some background to malaria itself. I know that some of you are probably familiar with it, but maybe not all of you, and then what we're trying to do specifically in modeling the development of malaria within the mosquito itself. And I just want to start off by saying all of the work that I'm talking about today is joint work with Olivia Prosper, who probably many of you know here. It's in the math department here at UT. So to give a little bit of background, when I talk about malaria, I'm going to be talking about Plasmodium. Typically, we think about Plasmodium falciparum, which is the main human malaria. All of the malarias are transmitted by a particular type of mosquito, the Anopheles mosquito. Some of the data that we look at today is actually a slightly different species of malaria that I'll talk about. Plasmodium falciparum itself is responsible for nearly 500,000 deaths, and about 50% of the world remains at risk. And it's a little bit hard to see here, but it's all of this sort of tropical area that we see here that remains at risk to malaria. So even though it's not really a disease that we hear about in the US a lot, there is a large risk around the world and a lot of people who this impacts on a regular basis. Malaria itself has quite a complicated life cycle. It involves two hosts, so a vertebrate host, we typically think about the human host, as well as the mosquito vector. And so what happens is an infected mosquito bites a human. It goes through a number of different stages within the human, including the liver and the red blood cell, and eventually it forms from sexually dimorphized stages called gametocytes, and if those are present in the blood and the mosquito bites, a mosquito can become infected. And then we get to the portion of the life cycle in the mosquito, which is what I'll be primarily focusing on today, what's happening with the parasite development within the mosquito. And so these males and females parasites come in, they can mate and form different stages, a zygote, an ookinete, and an oocyst. And then within the oocyst, these sexually mated parasites are going to undergo meiosis and produce haplotypes, and then those go on to be the stage of the parasite that goes to the salivary glands, and then the mosquito becomes infectious again. And so I'll talk more about this as we go through what are the aspects of that we're actually modeling and thinking about here when we're trying to understand how fast mosquitoes can go from this idea of picking up the parasites here from the blood and ultimately being able to transmit them onto produce another infection. So modeling malaria has a really rich, long history, over 100 years. Sort of one of the canonical models thinking about malaria is called the Ross-McDonald model, named after Ronald Ross and George MacDonald, who both had large contributions to that. It's sort of succinctly denoted through this compartmental model where we divide humans into this susceptible and infected classes, and we divide mosquitoes into susceptible, infected, or infectious. And you'll notice this distinction between being infected and being infectious, and that's because it does take this period of time within the mosquito to develop that, go through all those stages, and go from acquiring the infection to being able to transmit the infection, which we refer to as the latent period here. This compartmental model, there's been a lot of work done thinking about it in many different ways. One of sort of the canonical things that we talk about is the basic reproductive number related to that, or the average number of onward infections that a single infectious individual is giving us. And so that gives us an approximation of how the disease is going to spread. And based on all the parameters in that sort of previous compartmental model, there's this nice, simple mathematical formula thinking about what's happening here. And we'll notice that certain of these parameters are coming in a linear way, and certain ones are coming in a non-linear way. And in particular, Ronald Ross and George MacDonald noticed which of these parameters are coming in a non-linear way, and used that as a way to how can we best intervene against malaria. And so some of the ones that you'll notice come in here, this A squared, where A is biting rate, and mu, both in this exponential and in the numerator, the mosquito mortality rate. And so their recommendation was really, if we can shorten the lifespan of mosquitoes, we can really make an impact. And so this sort of simplistic idea using these simple compartmental models has had a lot of impact on malaria intervention strategies. And sort of the original malaria eradication campaign back in the 1950s was based on ideas from this model. But you'll notice here that one of the parameters of particular interest to us is this time to infectiousness. So for the mosquito, between when it becomes infected and when it can actually transmit that. And that also appears in the exponential here, so in a nonlinear way into our R0, into this basic R0 idea. And so we're going to be basically trying to think about ways to measure that and model that within the mosquito here. And so that's going to have an impact certainly on what we measure our R0s to be, although we aren't going to actually measure R0s in this talk. So measuring time to infectiousness, typically we think about this as the extrinsic incubation period. So throughout the talk, I'm going to refer back to that as the period between infection, when you acquire infection, until and that's going to be the ingestion of those gametocytes until infectiousness, and that's going to be when we see sporozoites in the salivary glands. We don't actually know at that point when those mosquitoes are going to bite again, but they are capable of producing infection, so we consider that to be the extrinsic incubation period. So throughout the talk, I'll have that abbreviation, EIP. And so here on the bottom is a schematic looking at the development of the parasite within the mosquito, so starting off with these gametocytes, or the male and female stages, all the way through to the sporozoites in the salivary glands. And then on the bottom, this is a characterization of the rough numbers that we're seeing. And you'll notice, maybe a little bit hard to see, but on the y-axis, we have a log scale. So we start off with something on the order of maybe thousands or 10,000s, and then we see this decline all the way down into the, dozens of parasites, and then we see this really strong exponential growth again during that production of those haplotypes within the oocyst stage. And so that's a very strong exponential growth that happens. They burst from the oocyst, and then they migrate to the salivary glands, and that's where they become infectious. And so we're going to work to quantify that with a mathematical model that we're seeing. So I mentioned that when we talk about malaria, we often talk about the species Plasmodium falciparum, but we're actually going to work with data that comes from Plasmodium berghei in the Anopheles stephensi mosquito. And so in sub-Saharan Africa, we're primarily thinking about transmission of Plasmodium falciparum in Anopheles gambiae mosquitoes. The reason we're using this data is a couple things. There's a really nice, rich data set that we're working with. It's coming from a thesis by Emma Dawes at the Imperial College London. And so parts of that thesis, including her experiments, her original experiments, are in published papers, but the data that we're working with actually has not been published elsewhere, but it is publicly available through her thesis. And so what they do in these experiments is that they're sort of skipping the initial stages that are happening here, the gametocytes and the fusion to form a zygote, and they're starting with that second sort of stage that I talked about, the ookinete stage. And I will note that I mentioned that this is a log scale in terms of the number of parasites. This is actually a very nonlinear scale in terms of the timing at the bottom. So this first tick here is 5 minutes. We get up to about an hour here, and all the way over to 24 hours. So they're sort of skipping this initial set of things, the initial about 3 hours that are happening. And the reason that they want to do that is there's a lot of things that are happening here that can change the numbers, can change the genetics that we're working with, and they wanted a very clean starting point. So they're starting a little bit later with these experiments by introducing ookinetes per microliter, and then they're going on to measure the number of oocysts, so that stage before the exponential growth, and then the number of sporozoites. Although they don't actually count the number of sporozoites because it gets very large, they sort of categorize them based on that. And I will note these numbers are for Plasmodium falciparum, and we're talking about a different species, Plasmodium berghei. So the numbers themselves are a slightly shift in the order of magnitude, but the trends that we see are still reflected with that species that we're working with, or they're working with. So in this data published in her thesis, there's these really rich experiments. And so I'm giving you a sample here of what some of the data looks like. And so on the left, we have oocyst data over time. And here we're back to very standard axes of days and number of oocysts that they're measuring. And each dot here is going to represent a single mosquito. In order to measure it, they have to kill the mosquito. So none of these points are repeated mosquitoes that we have. The sort of larger dots, or the squares, are going to be the average of those values. And then on the right, we have the sporozoite score, and they're going to measure that and give it the score between 1 and 4, where 0 means there are no sporozoites seen. 1 is going to be this 1 to 10 sporozoites, 2, 10 to 100, roughly 300 to 1,000, and 4 greater than 1,000 that we're seeing here. And again, you can see all the individual dots are individual mosquitoes. Again, the sampling requires the killing of the mosquitoes, and it's actually really hard to see where the averages are on these plots. And you'll notice that a lot of these points are overlapping as well. So this is the data that we have in graphical form, not in actual tables or anything, so we have to extract that data. And so we actually don't use all of the individual numbers, we just use the averages. And so let me go back for a second. The data is here, that's in squares. We can see it represented again here in the triangles, and then the lines that are on these plots are some modeling that they did themselves. And so in fact, they performed 3 different experiments. In each experiment, there were 3 different conditions, although experiment 1 and experiment 2 were repeats of each other. So there's actually 9 individual data sets that there are 2 that have, there's a set of 3 and a set of 3 that duplicate the parameterization or the conditions, and then a separate set of 3. So in total, we're really looking at 9 data sets here. Or the top row here is that experiment 1. Experiment 2 was a replicate of experiment 1, and so each of these is a different starting level of the oocinites. And then in experiment 3, they did a separate set. So we have 6 different sets of conditions that we're looking at. And again, the data here, the averages of the mosquitoes, and then the lines are some of the modeling that they did. So we extracted the data from these plots where we can actually see all the points, but keeping in mind that they are averages of the mosquitoes that are there. And we have that data for the oocysts, and we also have it for the sporozoite score. We have no data on the actual number of sporozoites, just sort of that log separation of the different types that they're working with. And again, we extracted this data that's graphically available to work with. And so they actually did a fair bit of modeling, unpublished. Again, this is all from within the thesis that they worked on. And they looked at mainly looked at 2 different models to try and, which is these lines that appear in the data that I'm showing. So the first model is going to relate to the lines in the oocysts, and the second model is going to relate to the lines in the sporozoite. And so just briefly to describe, in the first one, they look at sort of multiple ookinete stages and then the production of oocysts. So they're only looking at the oocysts that they have. And in the second model that they looked at, they started at day 10, looked at multiple oocyst stages, and quantify the number of sporozoites. And you may have noticed on those graphs, they had pretty nice fits to the reasonable fits to the oocyst data and reasonable fits to the sporozoite data, done from 2 different models. If you tried to employ either of these models to do both the sporozoite and the oocyst data, it doesn't fit well at all. And so we were ultimately interested in having a single model that could really fit all of the data together. And we knew that these 2 starting choices were not going to be able to, and we can show that I don't have that data here, that isn't going to work. And so what we did is we started out with a differential equation model. Again, we only have data from oocinites, so we're starting at oocinites, we're looking at oocysts, and we're looking at sporozoites. And we considered a couple different model structures, and then we considered the parameterization quite in depth as well. And so that's what I want to talk to you about today, and sort of the results that we fit in terms of the extrinsic incubation period, or that time to infectiousness. So we started off with a number of different models that we considered, but ultimately we sort of settled on 2 that we thought were pretty relevant, and I'll talk to you the main difference between them. And so here, when we start off with our level of oocinites in E, and then we assume that there's 2 types of oocysts that can be produced. Ones that are ultimately going to burst and produce sporozoites, and another sort of set that are not going to ever burst, but they may be subject to mortality over time. And so the reason we did that is because we see in the data that bursting happens somewhere between, I should say sporozoites appear somewhere between about day 15 to day 25. We don't see appearance of new sporozoites after day 25, but we still see the persistence of oocysts up to about day 40. 42 is when the experiments end, and we still see oocysts. So clearly not all of them are bursting, although when you look in the literature, no one really considers this idea. You start to talk to entomologists working with mosquitoes, and they're like, yeah, sure, yeah, of course, there's some oocysts that are staying around for long periods of time. So we chose to split it in this way. And then the main difference between model 1 and model 2 is going to be what happens in those oocyst stages. Here in model 1, we're considering linear transition between the stages, and we allow the total number of stages of which we have that linear transition to change. And so that's one thing that we're going to consider fitting. Whereas in model 2, we actually use a time-dependent bursting function. And so we have two parameters into that function that we need to fit to try and understand how that's actually happening. So this is going to have sort of a gamma distributed idea. This is a particular functional form. Now, this functional form comes from previous work that Olivia and I have done together, which is based off of other work modeling the development of the parasite within the mosquito that looked at a sort of stepwise time-dependent function. And we fit this as a very similar time-dependent function that didn't require a piecewise component to it. And so to give you an idea, the main difference here is in this bursting time. I'll talk to you about all the different lines here, but on the top we have our probability density function and our cumulative density function that we're working with here. The dashed line is our model 2, and so actually that's not a true CDF, but it's going to be what the time-dependent bursting function looks like. And we want to see if that our model 1 with a different number of compartments may be able to approximate that pretty well. And so as the lines go from darker to lighter here, the solid lines go from darker to lighter, this is keeping track of the number of compartments we have, so that N here. And so we're going from 2 all the way out to 100, and you'll notice that as we increase the number of compartments, we're able to approximate what's happening in that time-dependent bursting function even better. Although you'll notice that it's never identical to what we're seeing there. These vertical lines are going to be the 50, where we cross that 50% level. And so we can see that for a small number of compartments, starting with 2 over here, that's happening much earlier. But as we increase the number of compartments, then we're approaching where that happens with our time-dependent function. The idea being that these models should be relatively similar under certain choices that we're looking at. So I put up before the sort of the box and wire diagrams of what's happening. This gets translated into 2 different sets of ODEs. On the left, we have model 1. On the right, we have model 2. So it's a little bit easier here now to see that time-dependent bursting function from our oocysts going into our sporozoites, whereas here we have those N different oocyst categories that we're working with. And you'll notice there's a number of parameters that come into play here, and we're ultimately going to fit some fraction of those parameters. So what we have in our our ookinete compartment is that some of them die and some of them transition into our oocyst compartment. Some of those transition into the bursting oocyst, that's our parameter F. Some of them transition into our non-bursting population, so that 1 minus F. And then we only consider mortality in our oocysts that do not burst. That's coming up in this mu sub 0. And then we have this transition rate beta between each of our compartments. And then we have 2 parameters, K and T sub B, that govern our bursting function. And then ultimately we have 2 parameters, P and M, that are going to deal with the probability that sporozoites reach the salivary glands and how many sporozoites there are per oocyst that bursts. And so we actually, like I mentioned, there's a number of parameters that we have here The bottom two, the probability and the number of sporozoites per oocyst, we don't actually fit those, although they appear in both models. And then in model 1, we have 5 parameters, the top 5 parameters we see here that we fit. And in model 2, we have 6 parameters that we fit. So the first 4 here, plus the K and TB that are dealing with that time-dependent bursting function that we're looking at. And so when we do the fitting, we form our likelihood, and we use a Poisson likelihood for both the oocyst data and the sporozoite data. We don't actually do any weighting between them. We consider both of the components here, although we are using the sporozoite score, not the sporozoite number that we're working with. And so our lambdas here are going to be our model-derived outputs. Our O and S of I are our data, and ultimately we're going to look at our negative log likelihood as the combination, the unweighted combination of our likelihoods for our oocyst and our sporozoite. And then we fit them in a number of different ways. So we fit both Model 1 and Model 2 in MATLAB using the multi-start function, which means that we have a number of different starting points that we're looking at that are randomly chosen within some sort of interval that we give them. And this is a fast and easy way to look at multiple different models, but we're getting a single local optimum out of that. And then for Model 2, we also looked at using MCMC in R using the DEB infer package, which is specially optimized for ODE systems. But this requires a lot more time, especially if we wanted to look at large dimensional systems of ODEs. So in particular, that's why we did not do this with Model 1. Because Model 1, remember, when we start to have n compartments, then we have 104, 103 ODEs that we're looking at become sort of intractable for what we wanted to do. So we only looked at Model 2 here, which only has the 4 compartments, but has that time-dependent bursting function. And that gives us a full posterior distribution of all the parameters that we're interested in. So we can see a little bit about the variability that, of the fits as well. We know from before we even start that there are going to be some strong relationships between particular parameters that we have, just how we've set up the model. So going in, that gives us some information on how to constrain some of the parameters of interest. So first of all, we know that there are 2 ways to leave the ookinete stage here, and that's either through death or through transition into the oocyst compartment. So obviously there's going to be some correlation. If we increase death, then we may have to increase our transition rate to get the same number of oocysts that from a similar ookinete starting population. And additionally, we know that the population of non-bursting oocysts is going to have a high correlation with F, or really 1 minus F here, the fraction that are transitioning to that non-bursting compartment, as well as the death rate coming out of it. So the in and the out can be balanced. If you increase the input and increase the output, you can get the same number out. So we know that we expect there to be this level of correlation there. I showed you all of that in Model 1, but the same parameter combinations come up in Model 2. And we see these correlations come out quite nicely in our results for the MCMC, and that's not surprising at all. We also have a more complicated parameter relationship between our 2 parameters within our bursting functions. Remember, K is the numerator in that bursting function. That's going to give us the maximal bursting rate, and TB is going to tell you when that bursting function really changes its concavity that we're looking at. And so we can actually find we can analytically find when the time when the bursting population is half its initial size through this calculation here, and that's going to come up in our extrinsic incubation period calculation later, but we really didn't have a good idea of what K and TB should be. Those were things that we couldn't look at in the literature. There's no information on like what that bursting function is going to be. So we played around with thinking about what those dependencies are. And so here I'm showing you a plot of K between 1 and 20 and TB between 10 and 50. And remember, I I briefly mentioned this, but the experiments themselves went out to 42 days, so obviously we can't have a TB beyond 42 days that we're working with. And we can see that when K is greater than 1, we have sort of a slight correlation with TB, and then the values go up significantly here. When K is below 1, we see sort of a downturn here, and so we know that there is importance and connection between those that we're working with. Ultimately, we ended up bounding our K between 0 and 10, because out here, sometimes when the fitting hit K equals 10, it would just keep pushing as high as K as we let it go. So to give you an idea, the prior distributions and parameter bounds that we used in our fitting methods. So the top 6 parameters here are the parameters that are fit in both our multi-start in MATLAB, which gives that single optimum, and in our MCMC that we're working with. In the MCMC, we used uniform prior distributions with the bounds that we gave here. For mu and sigma, we kept a bound of 2 days because there is some experimental evidence to say that the loss of that uokinates is happening at a combined rate of 1 over mu e plus sigma e at about 2, but that's for a different species. And so we bound both of these by 2 here. And for consistency, because we didn't have any idea about this oocyst loss rate, in fact, many studies set that equal to 0. We bound that also between 0 and 2. And you'll notice in the results that it comes out quite low. F here is that probability of whether you go to a bursting oocyst or a non-bursting oocyst. We bound that between 0 and 1. And then I mentioned we have these 2 parameters, K and TB, that we don't have a good way to think about. We bound K between 0 and 10, and TB between the bounds of the experiment, 0 and 42. And then down here, beta is not actually in. Beta comes up as the average time that, or 1 over beta is going to be our transition rate between compartments. And so that doesn't come up in our MCMC, which is why it's not in a uniform, but we did use a consistent bound of 0 and 10 because of its relationship with K in the other model that we're working with. And so when we're thinking about choosing what is the better model to work with, we also have to make a choice within model 1, how many compartments, what is the best number of compartments to look at, and then choose between model 1 and model 2. So the first thing we're going to do is, using that multi-start in MATLAB, the single optimum, vary the number of compartments that we're looking at and see how that fits the oocyst data and the sporozoite data simultaneously. And so on the left-hand side, we have those oocyst data. We have 6 of them here. The reason we have 6 is we combined the data from the conditions that were identical. So we started with 100 uokinates. There were 2 experiments that actually had that condition, and so we put those together and fit that joint data set. And on the right-hand side, we have the sporozoites, where we've transformed them to sporozoite number. So we follow the sporozoite number in the model itself. With the data, we raised the value of the sporozoite score to the power 10 that we're working with to fit that. And so the different colors here, from this dark blue to a light orange, are going to be model 1 with different numbers of compartments. And so you'll notice that for many different number of compartments, we fit well in a lot of cases, and then we clearly have some that are just not fitting the data quite well in the oocyst compartments. Those ones that don't fit quite well typically are these higher number of compartments that we're working with. In contrast, on the sporozoite side, many fewer of the curves are going to fit the sporozoite data well. In fact, many of them start off much too early to rise, well before we see any sporozoite production at all. So you'll notice that most of these, nothing is happening until about day 10, and then maybe day 12 or 14 we start to see a few, and then by day 20 we see a lot, and it's kind of consistent in that level that we have there. And so many of these, fewer compartments, 2, 3, 10, 20, they're predicting the appearance of sporozoites much earlier than we see. And then we also have in red model 2, and you'll see that that tends to fit the data quite well in a lot of cases, but comparatively well to some of our model 1 fits as well. So in order to compare between them, so I'm plotting the AIC criterion here. So we want to be able to penalize for, remember, model 2 has an additional parameter, and we want to be able to compare them. And so for each of the 6 conditions that we're looking at, the blue lines and dots here are going to be these optimum fits that we have on that I just talked to you about. And then the black dotted line is model 2. So it's going to be that comparison. And so you'll notice that sometimes under certain conditions, model 1 never does as well as model 2. And then in some other conditions over here, it's getting close or just a little bit better in that fitting that we have. And this again is all the multi-start fitting that we're working with in MATLAB. And so if we want to compare the fitting, the sort of the best fitting that we found from our multi-start with the types of fittings that we found using just model 2 in our MCMC. So I've plotted those here. The black, again, are all the dots. On the left, we have the oocyst numbers. On the right, the sporozoite scores I'm showing you here, not the actual sporozoite numbers. And the red, solid red curves are our best multi-start, and that might be either model 1 or model 2. I'll show you some of the results deciding between them in a little bit. And then the dashed blue is going to be the median posterior from our MCMC. And you'll notice that it's really hard on most of these to even tell that there are 2 lines on those plots, because we're getting a lot of agreement between them. There are a couple where we see a slight difference, especially in that timing of the sporozoite uptick that we see. But we're getting a lot of consistency, as we would expect, between our multi-start fittings and our posterior median from the MCMC. But one of the nice things about the MCMC is it not only gives us that sort of median value, but it gives us spread as well. So this is the highest density posterior interval for our oocysts and sporozoites, again using model 2 in the MCMC setting. And so going from dark blue to orange is going to be an increase in the number of ookinetes. And you'll notice that if you look here, we don't actually, this is in order of smallest to largest, we don't actually see the same ordering here as well, right? So we see a little bit flip in the ordering between these 2 conditions here. Well, we see higher peaks, obviously, of the number of oocysts as we increase the number of ookinetes that we have, and also a shifting later in time of those peaks, which are all things that biologically we would expect. If there are more entities there, it takes a little bit more time to produce all of them before they can decay away. You'll also notice that we have decay, strong decay of the oocyst numbers, but nothing is really going all the way down to 0. So that's that persistence of oocyst all the way out in the experiment. That was to day 42, but here we're looking up to day 50. The other thing that you may notice is a slight kink in these curves that actually comes up in these curves as well. They're not just exponential decay. And what's happening there, that's the time at which the sporozoites, those bursting oocysts, are bursting to produce sporozoites, and they're shifting that curve ever so slightly. Then on the sporozoite side, we see in the MCMC that there's actually a pretty similar timing at which we start to see the appearance of the sporozoites, but some difference in the growth of those sporozoite curves dependent on the level of the initial conditions that we were looking at, and some differences in the variability. So these lower ookinete starting values actually have more variability, tend to occur slightly later, and have a lower slope, as you might expect. This really narrow one here, that's our largest initial condition that we're working with. And interestingly, it's a little bit hard to see, but it actually starts a little bit later, which is something that you we may not have predicted that with a very large initial condition, we're actually seeing later appearance of sporozoates that we're fitting to. And so, looking at all of our fitting, we can take a look at all of our parameterization. I know this is a lot of numbers up here, but I'm going to highlight a couple of them to look at. Each sort of block here is a different initial condition that we're working with, and then the two rows that say multi are our multi-start fits, our optimums in MATLAB, and it's a little bit subtle, but the ones that are bolded tell you whether model 1, as in the first two, or model 2, as in the next two, were the better of the multi-start fits. And then the MCMC is that final line in each condition. And of course, it's always model 2, because that's the only thing that we fit with the MCMC. And this number n over here tells us what is the optimum number. So when I showed you that AIC graph before, which of those dots was the minimum for model 1 that we're working with. And so if you kind of look across here, we see a fair bit of consistency between parameterizations, specifically between our Model 2 and multi-start and the MCMC, which also uses Model 2. I will highlight a couple of things here. First of all, we see this consistency in that T star, which is, remember, calculated from our T, B, and K, our parameters in our bursting function. T star is going to be really related to our extrinsic incubation period. We see quite a bit of consistency across, among that, and in particular, we see more deviation, like down here, where this is off by about 2 days, when we actually fit better with Model 2. So this Model 1 is, even though it's the best fit in this case, it's actually not fitting as well. The other thing I want to highlight is the value for F here. So F is the fraction of oocysts that, or sorry, the fraction of oocinites that go on to become bursting oocysts. And these are all very small values, on the order of 1, 2, 3, 4, 5%. So we're predicting that only about 5% or less of oocysts or oocinites go on to be oocysts that ultimately burst. So most of them are in that sort of non-bursting category, which is in contrast to what other, well, no one's really looked at that number before, but no one has made the suggestion that many oocysts are not bursting. And of course, in this figure, anytime that we have a dash, it's because that parameter doesn't appear in that model. I also want to highlight, remember, we didn't have a whole lot of idea of what this death rate in the oocyst compartment was, and we're seeing, I told you that a number of people have set that to 0 before, including ourselves in some previous models, but it's actually a quite low value. And then we had this assumption that these values were between sigma e and mu e to get combined were between 0 and 2, and we, of course, we restrict each of them individually between 0 and 2, but overall we generally see them, the sum of them, to fall between 0 and 2, or very close to it at least. We also took a look at the parameter distributions from our MCMC by each of our parameters individually, and there's some really nice trends, interesting trends that we see here. We've also, so the sort of violin plots, which have no color here, and the black lines are the medium of the violin plots, those come from our MCMC fitting. The blue circles are going to be our multi-start fitting from Model 1, and the red star is the multi-start fitting from Model 2. And so for certain parameters, we see a lot of consistency between all of the fitting methods. These do pretty well. For our initial ookinete, or lower ookinete densities, the multi-start is fitting something a little bit below the median, but certainly well within that distribution. We have other parameters where that's where we don't see quite as good fitting. For example, the mu e is there's a lot more variability that we see, and we also see a lot of sort of differences in between what's being fit with Model 1 and Model 2 that we have here. We also see something really interesting that came up in our fitting for F and our oocyst mortality, and that's the sort of fitting of this sort of up-down sequence. As we increase the number of oocinites, we see something that seems like not like a clear trend that we're looking at. And if we go back and look at it, actually, every other value that we're looking at here is with one experiment. So 100 uokinetes, 500, and 2,000 were all with a single experiment. And 200, 800, and 4,000 were with the other set of experiments. So it's actually, we're actually fitting something that's related likely to the experimental conditions, but not captured in a way, which we thought was really interesting. And so what we're seeing is like pretty good consistency, especially here, across a single experiment and across a single experiment, but not, no clear trend that's happening. And then in the bottom are TB and K. There are no blue circles there. It's not that you're just not seeing them. We don't fit those in Model 1, so we don't represent them there. And interestingly, even though there's some variation, in particular in our value for K at 500, there's a whole lot of variation. We're fitting things in the multi-start that are consistent with that. And one thing you'll notice with K here is a lot of the values are small, but a couple of them for the 500s and the 4,000 are actually sitting right at the bound. We forced K between 0 and 10, and remember, that was a somewhat arbitrary choice looking at those correlations between K and TB that we're looking at. And so somehow we think the variation is coming out in K and not as much in the TB. I will also highlight that a number of the axes here are constrained much more narrowly than the values that we allowed them to vary between. So for example, TB we allowed to go from 0 to 42. The axes I'm showing here are 12 to 24. So it's actually fitting something much narrower. The same thing with F and our oocyst mortality. So this F is that probability of becoming a bursting oocyst. We allowed that to go between 0 and 1. I told you that pretty much everything was below 5%. This is 10%, that's 5%. And over here, the mu0 was between 0 and 2, and we allowed it to go between 0 and 2, and, but everything was around 0.05. So the axes here are 0 and 0.1. The axes on these other graphs here for our transition rate to oocysts and our mortality of ookinetes and our value for our maximal bursting rate, they're at their full range. So we're really seeing much more variation in those parameters. So sort of to kind of summarize a bit of what I've already told you, we see a lot of consistency between the multi-start and the MCMC as we'd expect, but we get more information on the variability in the parameters from the MCMC, but we have the ability to look at a lot more things with the multi-start. There's a lot of variability in K. We don't have any biological way to restrict that we know of. And so we bound that between 0 and 10, but we're definitely hitting the edge of that bounds under certain conditions. We're suggesting a really low percentage of bursting oocysts. And when we first modeled it, we didn't include a compartment for oocysts that didn't burst, and we really couldn't fit anything well at all. And then we went back and looked at the data and realized, well, we do have oocysts that persist for a really long time, much longer than when the sporozoites appear or stop appearing with new sporozoites. And so that really led us to this idea that we should incorporate this percentage. And as far as we know, no one else has really looked at that. And we see this bursting of oocysts that's occurring between about 16 to 20 days. If you are familiar with other species of malaria, like Plasmodium falciparum, this is much later, but very consistent with the literature for Plasmodium berghei. So all of that's to talk about the model and the parameterization, but what does it mean for the extrinsic incubation period, or calculating that time to infectiousness? So we're going to calculate from our model this time to infectiousness as our T star value, and I'll go over what that means under the two models, plus 1 over the time at which you are in ookinete. So we are missing that about 3 hours at the beginning when you go from gametocyte to zygote to ookinete. So you can think about these values being shifted ever so slightly, but that at beginning time, which we aren't able to capture with this data because we start at the ookinete stage, is not included in our extrinsic incubation period. For model 1, we calculate T star as the time in that the cumulative gamma distribution reaches 0.5. So if you think all the way back to those curves, I showed you those vertical lines about where it's crossing 0.5. That's what we're calculating as T star. In model 2, we have this analytical solution. So once we fit our K and TB values, we can get a value for T star. So we can start to look at what do we predict the time to infectiousness to be as a function of our initial conditions here. And we generally see sort of a U-shaped curve, so that we're seeing shorter timing to infectiousness for intermediate values of uokinetes. And so I think the general expectation is that as we have more uokinetes, we should have a faster time to infectiousness. And we're actually seeing something that has sort of a limit in this intermediate level. And again, the violin plots are going to be from our MCMC, our posterior distributions here. The blue circle is model 1 with the multi-start, the red star model 2 with the multi-start. And we see the red stars fit really well because they're are very consistent with our MCMC. They're both fitting model 2, whereas our blue circles from model 1 are actually fitting something that's a little bit lower in terms of the time to infectiousness, or that extrinsic incubation period. We were interested, right, there are two components to our EIP, that T star and the 1 over sigma E plus mu E, and so we're interested in the contributions of each of those, and we could look at that as well. We see that T star is a large contribution and shows a very similar curve, but what we see is, by and large, except for this one condition, an increasing level time for as the uokinete number increases, and increasing time right at the beginning that we're seeing. And that sort of correlates with those later peaks in oocysts, larger and later peaks in oocysts that we were seeing as well. So we can start to sort of break down where these values are coming from. So ultimately, in this data, we fit an EIP that's somewhere between 17 and 21 days, depending on the uokinete density that we're looking at. We had much later times to infectiousness for our lowest value of uokinetes of 100, and our highest value of uokinetes, which is about 4,000. We see the lowest time to infectiousness for those intermediate densities that we saw. And we also saw, we predicted a lower time to infectiousness when we used model 1, which, remember, is that gamma distributed transition of between different compartments rather than the time dependent one that we're looking at. We also did a fair bit of sensitivity to this. We did it to many parts of the model. I'm only going to show you the sensitivity to the extrinsic incubation period based on the model. And we did this using extended Fourier amplitude sensitivity test, or EFAST. And that's going to vary parameters in a sinusoidal way, so we can use some Fourier analysis to look at the contribution of the Fourier coefficients. And we look at two different things: the individual sensitivity, and that's going to be the variance in these computed from the Fourier coefficients for a particular parameter versus the total variance that we see. And then we also look at the total sensitivity, and what that does is sort of lump all of the other, it focuses on a single parameter, lumps all the other parameters together, and looks at the contribution of just that single parameter, so you're able to think about higher order interactions with that. And so the individual sensitivities down here are in the orange colors. Lighter to darker is fewer to more uokinetes, and then the blue bars, lighter to darker again, are going to be the total sensitivity that we look at. On the left we have model 1, and on the right we have model 2, and then there's a dummy variable that we throw in as sort of a control that we're working with here. So anything that's on that level we assume is going to be noise. And what we see is, at least for the extrinsic incubation period in model 1, that transition rate between compartments is by far the most important. And in the model 2 we see those two parameters that inform the time-dependent bursting function are important, but interestingly, that timing of it, TB, is much more important than our maximal bursting rate, K. in this sensitivity analysis. We use the same sensitivity analysis to look at our time dependence of all of our different classes, our different compartments, and we didn't find anything that was too surprising there, which is why I didn't want to talk about it, but I'm happy to chat with anyone about that in terms of the sensitivity that we were interested in. So sort of to summarize what I've talked to you about today, we have this within mosquito model of the parasite dynamics, a little bit truncated. We don't include the first couple compartments. We use it to estimate the time of infectiousness, and then really think about what are the parameters of interest and how are they impacting that. And ultimately, those are things along the lines of where could we intervene in the mosquito to really make a difference. And another thing to highlight is that fraction of the bursting oocysts seems to be important and something that doesn't seem to be have noticed before. I, of course, want to put this with a number of caveats. We are skipping that early part in the mosquito where we know there is significant variation between the number of gametocytes and the number of ookinetes that get formed. That was the entire idea behind starting that experiment a little bit later. This is done for a particular parasite species, Plasmodium berghii, and a particular mosquito species, Anopheles defensii. So of course there might be differences for other parasite species mosquito combinations. And of course I want to highlight that our models didn't always match up. And so we are interested in looking at that a little bit more. But additionally, we're interested in looking at ideas and extensions of this. And one of the things that has come up is that is the number of oocysts actually the right metric to look at when you're measuring these things in mosquitoes? There's been some recent evidence that it may not be the number that's so important, but the size of the oocysts, and there's been a number of really nice papers that have come out recently. And so we are thinking about that as the oocyst number and size and number through a PDE model, and that's actually one of the things that I'll be around this week working with Olivia on. So hopefully we can see if there's a better way to think about these things or a different way to get more information about this as well. Before I wrap up, I just want to highlight something that we're running this summer, a American Math Society Math Research Community. It's a week-long research workshop for young career individuals. We're going to do it on the dynamics of infectious disease, ecological models across multiple scales. The deadline to apply is February 15th, and the website for it is up here, but if you have any questions, you're welcome to email me. And with that, thank you all for listening. I'm happy to take questions.
So thanks a lot, Lauren. So the question about time to infectiousness is of tremendous interest to people interested in the impacts of global climate change on mosquito distribution and potential for maybe changing malaria distribution. And so I have some vague memory. I don't remember the details. Matt Thomas and Ottar Bjornstedt looking at issues of temperature variation.
Okay.
And so correct me if I'm wrong. The experiments this study is based upon were constant temperature?
Constant temperature, yes.
So do you have any insight as to how much variation in time to infectiousness would arise from temperature variation? Admittedly, I think most of the data is not in this species of malaria. I don't know, but
yeah. So those
relative to the variation that you've seen in terms of control.
There is quite a bit of variation in the temperature dependent changes, but it also matters a bit on how far you take your temperature curves out. So a lot of I don't know if I'm specifically thinking about their data, but there is a really nice recent work with Aaron Mordecai's group and Sadie Ryan's group that also looks at temperature and might be based on some of this work as well.
Yeah, I think they're all connected.
Yeah, I think, yeah, and so yes, I would believe that. And they see these quadratic or Briere-shaped curves, so hump-shaped curves that are sort of pinned at, essentially there isn't parasite development because there isn't parasite survival at certain bounds, and those are pretty extreme, certainly on the low end compared to where a lot of the malaria is happening, but you could argue that sort of at the upper end, it would make a big difference. And so when you're near the top of that curve, I would say you're very much on the orders that we're talking about here. But when you start to tip over and get into those steeper components of the curve, then yes, those are going to be much stricter bounds. It actually brings up a really interesting question of could we incorporate temperature in this as well, and I think that is something that would definitely be worthwhile. I also want to add on there's some recent work that looks at other sort of internal changes to the mosquito, maybe changing the timing, the parasite development time, which is what other people have looked at with temperature, and have shown that there may be certain factors, co-infections, different things that also could change that. So I think thinking more carefully about what in addition would change those dynamics is really important. Traditionally, it's just been thought of as one number, like this time period. And I think all of these things are getting at that it isn't just one number, and it's dependent on a lot of factors. People have thought about temperature. Here we're thinking about sort of other components that could come in, and I think ultimately all of those factors need to be thought about.
Thank you.
I don't think I got the point of you had different initial conditions for so that's how they did the experiment with different number of oocysts starting?
Different number of oocysts, yeah.
Oh, yeah. So how does that correspond to the initial number of oocysts? And how is that matching to what it's in the field? Because it seems like there's very few oocysts, mosquitoes that are in the field. One. And second, how do you actually make exactly 200 okinets per mosquito, like experimentally? I'm just curious.
So I can't answer the second part of that because I obviously did not do that. My understanding from reading their methodology is they're measuring within a microliter, and so it may not be an exact number, but it's going to be, they are actually counting what they see in those.
So I wonder how much error is there in that process, just because of variability you see in the data. I'm really and so is that all that noise maybe coming from that, or? Yeah.
It's potential. So let me answer the first part of your question too. So they are starting from the ookinetes. The ookinetes transition into oocysts, and so that's actually a relatively short process, and there is known to be loss because you have to cross the midgut wall, and so that's not something that not all ookinetes are able to attain. But other than that, there doesn't seem to be any sort of there's studies showing that there's not really any evidence of competition. There's not any evidence of like predation or something within the mosquito or immune response, but just that physical barrier is doing it. So the ookinetes seem to be very well correlated with, our oocyst starting condition is 0, but we see that transition, and so that you're probably thinking about like the peak level or something seems to be pretty highly correlated with the ookinete number that we see. So in the field, typically oocysts are, I will put this in a caveat. This data is Plasmodium berghei. I do not have any knowledge on what happens in Plasmodium berghei in the field. If you're talking about Plasmodium falciparum, yes, field study is much lower than typical oocyst density, like the 10, 20, that's like high. In the lab, a lot of the lab work works with oocyst numbers that are much higher for a couple of different reasons. Just the ability to have data and know that, but also they get these higher density experiments quite often. There are some questions about whether those exist whether there's higher ones that exist in the wild and we're just not seeing them. And I know a number of the people that I work with in mosquito labs really feel like this sort of maximum of 10 is too low, but it's probably not as high as what we're seeing in the lab. We're just not measuring it. But that's, there's no evidence that's saying that. But again, that's all Plasmodium falciparum. I don't know what field would actually be like for Plasmodium berghei. Yeah, it's more of a lab system that people work with.
About sensitivity. You know, it
Yeah.
seems like you have a lot of graduate students in the audience and postdocs, and it seems like now people are using, you know, a variety of, people used to use PRCCs, people are using, you know, Sobel, and you're using EFAST. Do you have any advice about that in general? Which types to use or anything?
My advice, not being an expert in that area, but would be that you want to look at what the data is, what your data is, and sort of correlations in it. So certain types are going to obviously only work under certain conditions or correlations between your inputs and your outputs. So that's a starting point, is making sure that you are using a type of sensitivity analysis that is consistent with what's happening there. To be honest, this was the first time that I've used EFAST before, and I'm not sure we needed to use EFAST here, but I was interested in exploring it and trying it, and because we actually don't see huge nonlinear correlations between our input and output, such that this would be a really nice choice in that direction. One of the things that I will say that we did run into in using that sensitivity analysis was that obviously the bounds of our parameters that we allowed in our MCMC was much larger for certain parameters than really the variation that we were seeing. And if we allowed those same bounds in doing a sensitivity, we also saw that those parameters were really sensitive. And so when we originally ran the sensitivity analysis using the same bounds, we went, oh, okay, this is kind of nonsense. And so I do caution you to think, not just sort of use the method, but think a little bit about what is it like. These methods are only really as good as what you're giving into them as well. So.
Well, also, like with multi-start, you know, the question is, you know, you have a certain number of parameters to estimate, and how many multi-starts to do, whether you do 1,000 or, you know, that's another thing. Maybe you have advice about that also.
Yeah, I feel like that's where you're starting to get into the art part of mathematical modeling rather than the science. I'm not sure I have you should do this every time, but I think you need to think critically about what you're getting out, and, you know, do things make sense? And if you change them, do they change? And, like, you know, just don't run it once and think that's like it's all solved. But yeah.
Hi. I'm just wondering if it's just the way how you incorporated the time delay is the difference between the two models, 1 and 2.
Yeah. So the
second one is I just didn't follow the second one, the functional formation. Could you just explain?
Yeah.
Thank you.
Actually, let me go back. This is going to be a while. Yeah. So I went through that a little bit quickly, but yeah, our functional form here is this constant in the numerator, and then this 1 plus the exponential in the denominator. The two parameters we're fitting are the constant in the numerator and this TB, which is going to, so to go back, it's going to tell us where this shifts concavity here. That's the TB, and then the KB. The K is going to tell us the height, obviously not of the CDF, but of that function itself that we're working with. And so the TB tells us where that curve shifts, and the K is telling us the height of that curve that we're employing as a rate. Yeah.
From the gamma form.
So the difference is, so let me go here. So our other form that we're looking at is transition rates between n different compartments that we're working with. So like this picture here, these transition rates between those compartments versus that time dependent. And that's exactly what we're showing here, that the bursting is happening. The dashed line is that Model 2 time dependent function. The solid lines are what's happening for different numbers of n's that we see here. And because of this formulation with these linear transitions, we can keep beta the same, but as we multiply it by n, when there's n more compartments, each individual step is faster, but the overall time length is the same, the average time length. And so that's a standard transition that comes up in sort of infectious disease models that allows us to shift from an exponentially distributed constant rate to a gamma distributed period. Yeah, that's going to be dependent on it. And that's why it's a great question, because I actually use beta n as my transition, because the transition between each individual stage, but the overall between stage 1 and n is going to be consistent between all of the models with the same beta value that we have. But you'll see that bursting happens really early when we have very few compartments, and we can get it to happen at a very similar time to the time dependent, but not identical time to the time dependent that we're working with. Thank you.